All right, we will go ahead and get started, everyone. I want to just welcome you all to the TERN webinar, Nature's Litter, Nature's Treasure. Thank you so much to everyone for being here today. Um, we've got a lot of people joining us, over 300 from across Australia and around the world. And I just want to begin with an acknowledgement of country. We'd like to begin by paying our respects to the traditional custodians throughout Australia. We pay our respects to elders past, present, and emerging, and acknowledge the important role that Aboriginal and Torres Strait Island people continue to play in our community. I'm joining you today from Yagara and Turrbal country, located in Brisbane, Queensland. And for those who haven't met me, my name is Fionn McMillan Webster, and I am the science writer and content strategist at TERN, Australia's Ecosystem Observatory. So, first, a little bit of housekeeping. Please feel free to ask questions throughout the webcast using the question and answer function at the bottom of your screen, and we will answer them toward the end. If you have any technical issues, pardon me, comment in the chat function and we'll try to resolve them as soon as we can. You can also use chat to connect with other attendees. We are recording the webinar today so that we can share it with those who could not attend the webinar live. All right. So, a little bit about TERN. For those who are not already familiar with TERN, we're a national research infrastructure project funded by the Australian government. We make all the observation and data collections that tell us about what's changing in the environment. We also collect physical samples and develop a range of tools and apps. And our goal is to help researchers studying both land and coastal ecosystems to gain a deeper understanding of what is happening, including both the magnitude and direction of change. When we say infrastructure, we mean our people, our instruments, and our data. They're all covered under the definition of infrastructure. So let's meet the speakers for today. Today's webinar is Nature's Litter, Nature's Treasure. Australian vegetation produces hundreds of millions of tons of dead plant material each year, and that number is obviously much larger when you take it into a global perspective. What happens to it next, whether it decomposes quickly or persists for millennia, determines soil fertility, carbon storage, wildfire fuel loads, and the accuracy of our national environmental accounting. Yet this fundamental process remains one of ecology's most perplexing puzzles, and it plays out differently across ecosystems. Today, we bring together leading experts to explore how we take vastly different processes in diverse ecological contexts and create coherent national models that actually work. It's my great pleasure to introduce our speakers. Our first speaker today is Professor Heather Throop, and she's an ecosystem scientist at Arizona State University in the US. She studies how carbon and nutrients cycle through plants, soils, and the atmosphere, with a particular focus on arid and semi-arid environments. She is going to give us a dryland perspective on the role of leaf litter composition in carbon and nutrient cycles. Our second speaker today is Professor Mark Hovenden. He's a plant ecologist at the University of Tasmania with a particular interest in plant interactions and how the behavior of individual plants leads to the function of the entire ecosystem. His research includes exploration of carbon cycling in peatlands, and he will speak about that to us today. And our third speaker today is Dr. Jackie England, who is a forest ecologist at CSIRO with expertise in the measurement and modeling of carbon dynamics in woody vegetation and soils, and the impacts of climate and management on these processes. She will be speaking to us today about developing models to predict plant litter decomposition and some of the challenges and opportunities in that space. So I'm going to ask Heather to turn on her camera and I will turn mine off and she will begin and we will catch up with you at the end for Q&A. Thanks, Heather.
All right. It looks like I just requested access to share. Okay. Thank you so much, Fiona, for the introduction. It's really a delight to be here today, and it's wonderful to have the opportunity to share this space with Mark and Jackie and the excellent work that they're doing. So I'm joining you today from Arizona, from the Sonoran Desert in the southwestern U.S. I find when I start talking about litter, I can sometimes get some confused looks. So let's just initially start off and make sure that we're all on the same page. Litter means a lot of different things to different people. Today, we're not going to talk about these kinds of litter, but in the context of this webinar, let's agree that we're talking about instead the ecological perspective on litter, which we define as dead organic material. So in most systems, the majority of litter is from dead plant material. So leaves turn brown and fall to the ground as litter. Or dead grasses can be standing dead litter. Until then, they might fall and become surface litter. So this is the litter that then is nature's treasure. And litter makes up a substantial amount of the biomass in many systems. So there's a really nice synthesis that Clark et al. Published recently that explores the impacts of litter on ecosystems. It focuses on grasslands, but it's broadly relevant. And they point out a vast range of ways that litter impacts ecosystem functioning, including altering the biodiversity of many organisms, while also altering water and nutrient dynamics. And I'll add that fire is a really key factor that is modified by litter, particularly in fire-prone ecosystems such as many in Australia and the southwestern U.S. So these changes in ecosystem functioning in turn then influence ecosystem services that the systems provide. And I want to highlight decomposition as an ecosystem service. So litter decomposition is really critical for meeting carbon cycling as well as nutrient release back into systems. So we start by focusing on the carbon cycle. So we have a wealth of excellent data and really great modeling that allows us to pretty well constrain the amount of carbon that comes into a system via photosynthesis. And we have good ideas of what the constraints over large scale carbon release from ecosystems through soil and plant respiration. But the story gets a little bit more murky when we get down to the litter pools and what controls the turnover of those. So during decomposition or the breakdown of litter, there's carbon can be released into the atmosphere, or carbon and nutrients can be mineralized and released into soil pools. And these processes are driven primarily by microbial and invertebrate decomposers. But we know kind of the basics of the processes, but we have pretty poorly constrained estimates of the controls and the rates of these processes. And so Mark, Jackie, and I will share some different perspectives on these controls and our approaches in different systems. My own work on decomposition is focused in drylands, so let's start out there. Despite their global importance, our knowledge of ecological processes in dryland systems lags pretty dramatically behind wetter systems. So, controls over carbon and nutrient cycling processes are particularly poorly understood in drylands. And this plays out certainly in decomposition. So we can look at decomposition models in wet systems, really quite simple models that are based on abiotic control. So things like climate at the large scale. These models predict decomposition quite well in music systems. They've worked this way well for decades. We can add a little bit of nuance in add some local variability by, say, looking at particular decomposer organisms or vegetation characteristics and improve the models so that they're really, really fantastic. But if we take these same models into dryland systems, they just don't work well, and that they consistently underpredict decomposition in dryland systems. So my work on dryland litter decomposition has focused on trying to understand, what is it that makes dryland decomposition different in these systems? And so I'd like to share some ideas with you, and as well as a new approach that we're taking to this problem. So we'll start out talking about dryland mechanisms. So I'm defining dryland mechanisms as a suite of processes that are uniquely important in drylands. Grinsweig et al. Laid out, I think, about 12 of these processes that have unique features that stem from environmental conditions, things like high temperature, light, low moisture conditions, as well as sparse vegetation in drylands, and that these lead to some unique factors in drylands. And for decomposition, there are three dryland mechanisms that seem particularly important. And so these are photodegradation, which is the breakdown of litter by UV and other wave bands of solar radiation. Then we have thermal degradation, which occurs from chemical bond cleavage that happens during high temperature conditions that can occur in drylands. And then soil litter mixing is the facilitation of microbial activity and the buffering of moisture and temperature conditions from soil and litter mixing together on loose surface soils. So each of these mechanisms seems to speed decomposition in drylands, and these aren't usually included in models. We have kind of limited data at this point on thermal degradation and soil litter mixing, but photodegradation has received quite a lot of research attention in the last 20 years. And so now we've got to the point there's some nice syntheses and meta-analyses of photodegradation. And these are really interesting because the syntheses consistently show that photodegradation can increase the amount of decomposition. But this mechanism accounts for a minority of decomposition. So in all of these studies, then a small amount, maybe up to a quarter of the decomposition is from photodegradation. And these are in studies that really are set up typically to optimize the conditions where photodegradation would occur. So dryland mechanisms seem to be part of the story, but likely the majority of decomposition in these systems is still from biological activity of decomposer organisms. So another thing that we've thought a lot about is the role of temporal heterogeneity or the variability in time with decomposition. So, like ecologists love to simplify the world, right? We always appreciate a simple relationship between something like a climate variable and an ecological pattern. And dryland decomposition has consistently evaded this kind of characterization. And so, for example, this figure highlights the lack of relationship between annual precipitation, so that's here on the x-axis, and decomposition on the y-axis, where we have faster decomposition up towards the top. So there's really no pattern here. Although in wetter systems, we typically do see that moisture is a controlling variable for decomposition. So this has led to the suggestion that, well, maybe moisture is not really that important in controlling dryland decomposition. But we see really different patterns when we look at different timescales. So in this study, we looked at we used a manipulation of soil and litter and changed the amount of water that was added into these little microcosms in a glasshouse experiment. And so we found that if we watered these jars that had soil and litter in them, we added water. So we changed the total amount of precipitation over a month, splitting that up into um, water that was added in equal amounts every day. And so if we increased the amount of water, then we had more decomposition and ended up having less litter mass remaining. So that seemed to make sense. But then the story got more complicated and really more interesting when we looked at data from when we had split this up into delivery of the water only every 4 days. So same amount of water, but delivered less frequently. And in this case, then it didn't matter how much water we gave. It was really sort of equal decomposition uh, throughout if it was in this infrequent pulses. And so the total precipitation didn't impact decomposition, but these infrequent pulses led to just as fast decomposition as the infrequent large pulses. So total precipitation was less important than timing. And we found this consistently that when we look at these coarse timescale averages, they have pretty limited utility in dryland systems. So we need to consider the context of the timing of moisture um, and particularly given that it's enabling microbial activity, and it becomes at longer time scales, it really becomes decoupled from the total amount of rainfall. This is really particularly the case with litter, given how quickly litter moisture can change due to environmental conditions, since not buffered, it's just sitting right there out on the surface. So let's take a look now at spatial patterns. Music systems such as this forest are relatively spatially uniform, right? And particularly when we compare that to the extreme spatial variability of drylands, these bare spaces in between plants. And so these bare patches lead to horizontal litter movement across the soil surface, with litter accumulating next to objects that resist transport. Litter movement is altered by factors such as the strength of wind or the size of objects blocking movement. And so then we end up with very uneven accumulation of litter. So it ends up in biopress or animal footprints or next to plants. But then we have a really big problem with the data that we have in the decomposition literature. The majority of decomposition studies focus on bare areas in between plants. But in fact, much of the litter is in areas, say, like next to plants or underneath plants. So it seems that where litter ends up really matters for decomposition. So if we look, say, under a shrub in this subcanopy environment below the shrub, then there's typically high decomposer activity. Lots of microbes are there, high moisture environment, low solar radiation, and so biological decomposition is quite high. In contrast, if we look in intercanopial areas away from plants, then there's low moisture, pretty high temperature and solar radiation. So the dryland mechanisms like UV photodegradation become really important there. And we can see this when we synthesize decomposition rates in different dryland ecosystems. Here we calculated decomposition as the total mass of litter lost annually on a per area basis. And this takes into account that both the amount of litter present and then how quickly that litter decomposes on a per mass basis. And so we see there's a big difference in the total, the numbers, the amount of decomposition between these three deserts. Um, but we also see that the deserts differ a lot in where the bulk of the decomposing litter is. So these colors are different locations within the different deserts. But then it's really interesting that we would reach an entirely different conclusion if we had focused only on those inner canopy spaces away from trees, but where most decomposition research takes place. So these will lead us to dramatically underpredict decomposition within the landscape. So we've identified a couple different reasons that that we have currently a pretty limited capacity to predict decomposition. And so how can we improve our approach? I'm really excited that I'm currently leading a U.S. National Science Foundation grant that has the rather ambitious goal of improving our characterization of decomposition from the leaf level to the landscape level in North American drylands. We have a really fantastic team of collaborators in the U.S. and Mexico. We're characterizing litter distribution, environmental variables like solar radiation, moisture that affect decomposition, and the decomposition rates at multiple spatial scales. And so we do a lot of field work, as well as then we use airplane and satellite based observations to scale those up. We're using a series of hierarchically nested models to explore the impacts of different driving variables and their heterogeneity on the predictive capacity of our models. And we're working at the dryland sites of the National Ecological Observatory Network. This is the US sister network of TERN. So we're really excited to have the ability to leverage the vast data sets that NEON has and to also contribute to better understanding of these NEON field sites. And we're really excited now to launch a collaborative network for dryland decomposition research. We'd love to have any of you join us with the DEAD network as we strive to better characterize decomposition in drylands. You can check out our website and join our mailing list to learn more about our efforts and help us understand nature's treasure in drylands. Thanks very much. And with that, I will turn it over to Mark and his view of decomposition in a really different system.
So thank you very much, Heather, for that fantastic introduction to the whole process of decomposition and what happens to litter and the definition of litter and what happens in a very different system to what I'm going to talk about. So I'm going to talk about these ecosystems that going with the idea that litter is the ecological treasure, that I'm going to be talking about ecosystems that are built on buried treasure. Oh, hang on. There we go. I'd like to start by saying that I come from the Palawa country of Lutruwita, Tasmania, and the work I'm going to be talking about today has happened predominantly on Larramanara land. It also has had the support of these marvellous partners, the Tasmanian Land Conservancy, the Australian Research Council, and it is part of what is the Australian Mountain Research Facility, which is a TERN infrastructure funded through the NCRIS. So these, what I'm going to call peaty ecosystems, we often think of as these massive networks of wetlands. But in Australia, those are quite rare and only cover very small proportions. People that work on peatlands like to use the 3 and 30 statement, which is peatlands only cover 3% of the terrestrial surface, but they account for 30% of all the below ground carbon in ecosystems. So they're incredibly important. In Australia, if we were to map the peatlands, they wouldn't be able to you wouldn't now be able to see them on any map that has a scale of the size even of a reasonably small area. That's because they tend to exist in mosaics only in very specific situations. Except in Western Tasmania, where there is the large World Heritage Area listed button grass moorlands, which are true peatlands and cover large areas. And this is a totally unique ecosystem. What I am going to talk about today are systems in general where this beautiful circuit of carbon cycling that Heather so nicely described to us, and Jackie is going to tell us in much more detail about and tell us how we can model this system. I'm going to talk about systems where this cycle of carbon dioxide being fixed through photosynthesis into plant biomass and then added to the soil as the treasure, as litter to form soil organic matter, and on its way back out to being decomposed back to carbon dioxide gas, where these systems, because the soils are saturated, and in many instances the soils are extremely cold, this process of decomposition is retarded. And that means we have the treasure buried in the system and accumulating in the system. Now, that means globally, we end up with these beautiful peatlands where you have peat to an incredible depth, and they've been harvested by people for a long time to use as fuel and for creating beautiful whiskey. But in Australia, these, as I've said, are incredibly rare. I'm going to speak now and tell you a story about one system that has very peaty soils, but does not qualify as actually according to the international global definition of a peatland. So Tasmania is the large southern island to the south, obviously, of the mainland of Australia. And throughout the central areas of Tasmania, we have these highland, what we call peaty grasslands. So these grasslands and sedgelands are existing on these organic soils that are very rich in organic material, but they are not true peatlands. And if we map those, even on a very small scale of Tasmania as a whole, you can see we have the true peatlands in the west of the state, where you have the more rich orange and brown colours. And this is showing the extent of organic soils. But you can see even if we focus in on a small area in the centre of Tasmania, which is much drier than the west coast of Tasmania, there are large areas of organic soils interspersed throughout the ecosystem and throughout the entire landscape. So these are an incredibly important part of the overall landscape of southeastern highland country of Australia. These grasslands have incredibly organic rich soils, as you can see here from this spadeful of soil, but it doesn't have the depth such that we saw of that person that was harvesting the peat in Europe. So we have soils that have very high carbon content in the upper 5 to 10 centimeters. And this is a graph showing at the depth profile of soil carbon. And if you want to turn soil carbon into organic material, you basically multiply it by 2.5. And so here where we have 20% soil carbon, you're basically ending up with 50 to 75% of organic material in the top 5 to 10 centimeters of these soils. Now, these high country peaty grasslands and sedgelands are incredibly important that throughout southeastern highland areas of Australia, they form a mosaic with forest and woodland. They are extremely biodiverse. The plant diversity in these grasslands is tenfold that in the surrounding forest. And I'm not exaggerating, it actually is tenfold higher in these grasslands, the diversity, than what you get in the surrounding forests. They are fundamental to the high country food web because these areas are where we have the high densities of both invertebrate and vertebrate herbivores. And so therefore they are feeding on the plants and they attract all of the carnivores. So the entire food web is built on the existence of these open areas where the animals like to feed. Even though they might shelter in the day, the vertebrates shelter in the woodland and the forest around them, where they feed is out in the grasslands. They also provide essential ecosystem services, importantly for us as humans, in water capture, filtration, and storage. They are very dense in carbon, and they're very important grazing and hunting land, so they're culturally important both to the traditional owners and to European settlers. The highland peaty grasslands actually depend on these peaty soils that they create. So not only does this system create the peaty soils, but its very existence depends upon the persistence of those peaty soils. So what would happen if we lost these peaty soils is the plants, the woody plants from the surrounding areas would invade, because this is an ecosystem where the abiotic conditions, the external climatic conditions are such that trees would always win. And that means if these systems were invaded by trees, you would lose the grassy nature, you would lose the species that exist there in the open, and therefore you would lose the entire function of the ecosystem from the herbivores, the grazers who require these herbaceous plants, and therefore the carnivores that depend upon them. So this system is totally dependent on the peaty soil. And the reason is that the woody plants, the seedlings, cannot establish in these peaty soils because the peat, the undecomposed plant material, locks up the nutrients. And because it's to a reasonable depth, 15 to 20 centimeters, before you get to any form of mineral soil, that prevents the seedlings from getting their roots into the mineral soil. They starve from nutrients. And also the fact that the grazers like to come out, the grazers will come around and knock their heads off. And so it's a system that relies on these peaty soils. So therefore, we need to know what their fate is in a warmer and drier world, because the only reason we have them is because decomposition is retarded by the fact that the soils are cold and moist. So if we have a warmer, drier world, we're losing that retardation of decomposition. So this is a story about some experimental work we've been doing over the past 12 to 14 years in a site called Silver Plains, which is a peaty grassland up in the Central Plateau of Tasmania, owned and managed by the Tasmanian Land Conservancy. In 2013, we established a warming experiment where we use these open-top warming chambers to give us exactly 2 degrees of warming and 1 degree of warming at 5 centimeters in the soil. And we had highly replicated. And what we did is we measured the carbon exchange of these systems throughout the year. So we had dozens of soil respiration campaigns year-round in a range of conditions, from lovely summer conditions through the winter and through times when the soil was flooded. What we found, we've done this for many years, but this is just one typical yearly trace I'm showing you. So what this is the respiration of the ecosystem, so that the emission of carbon from the surface of this ecosystem, from the grassy surface, so including the soil organisms, all the decomposition processes, and the plants themselves. And what you can see in the yellow bars here is the rate in the chambers, in the warmed chambers, and the blue bars are showing us the rates that we had in the unwarmed chambers. And it's quite obvious that we have an acceleration of the carbon emission from this system during warming. But it's not only because the system is warming. We found when we plotted these against soil temperature, so what we have here is again the same variable, the efflux of carbon dioxide from this system, as a function here of soil temperature on this x-axis, but also with soil water content, shown in the size of the circle. What you can see in the unwarmed, in both conditions, as temperature increases, you have a very fast rate of increase of CO2 efflux from this system. But what you can also see in the dark points and the dotted line is that at any given soil temperature, the emission of carbon from these systems is accelerated. So warming is stimulating carbon loss directly from this system by making the soils warmer, but it is also stimulating carbon loss from this system by altering the dynamics of carbon emission, by actually changing the processes, the underlying processes of carbon cycling in this system. So we know that warming is increasing the risk to these peaty soils. What we don't know yet, well, what we didn't know, was what the background rate of this system is. These peats have been dated in nearby big peat bogs to be between 8 and 10,000 years old. So we know that they have formed since the last glaciation. But we don't know whether they are still accumulating carbon or whether we are just slowly burning through carbon that has accumulated over thousands of years. So we need to measure the background rates of carbon cycling. And this is what we've been doing for the last 6 years through the TERN-funded flux tower system that we have as part of the Australian Mountain Research Facility. And you can see here in this picture one of our flux towers. When we show the carbon balance annually, this is over 6 different years, and you can see this is the carbon balance starting on the 1st of September, where we go from the 1st of September, which is the beginning of our spring in the Southern Hemisphere, through to the end of winter, the following August. You can see that what happens is that a negative number here means a carbon gain in the system, and this is the cumulative carbon balance of the system. So the system, as it accumulates carbon during late spring and through the summer, the system is on balance since the beginning of September, a net sink. And at some point, that system stops being able to accumulate the biomass in the system, and its loss through carbon emissions through respiration starts to exceed the daily rate of carbon uptake, and we start returning back to zero. What you can see is enormous variation from year to year. In 3 of these years, which were wet years when we were in a La Nina period, you can see that this system at the end of the year was accumulating peat. So this treasure was being buried by the end of the year, and there was more treasure being buried than was being lost. However, in 3 other years, the only other 3 years we've had so far, all of which have been drier years, but not La Nina years, so these are just drier, normal drier years, we have on balance a total loss of carbon from the system. So we are burning through the peat. The other thing is you can see that in any one of these years, there's so much carbon burnt that we immediately wipe out the gains that we made from 3 years. So on balance, this system is at the moment losing carbon. So the subalpine grasslands that I'm talking about can accumulate peat, can accumulate this buried treasure, but only in the wet years. So this system, which totally depends upon the accumulation of plant litter exceeding its loss, and so therefore the peaty soils, depends itself upon the soils remaining wet. So the questions that we're asking now are, how wet do the soils need to remain and for how long? And that's what we're doing by installing a whole lot of new facilities and actually measuring the linkage between hydrology and the carbon balance of our system. So thank you very much. I'd now like to pass on to Jackie, who's going to give us a much better overview of the actual mechanisms that are controlling decomposition and litter cycling in both the wet systems I've been talking about and the very dry systems that Heather was speaking about.
Thanks, Mark. That was fabulous. I'm just going to share my screen. Okay, I'm hoping that's come up okay. So thanks, Heather and Mark. We've heard about two very contrasting systems in regards to litter and litter decomposition. I'm actually going to take a slightly different view. I'm going to provide a more generalized overview of how litter decomposes, including the phases and controls on decomposition. Describe some approaches to modelling and some recent model developments that capture the dynamics and drivers, mostly focusing on carbon, and highlight some applications of the models. So we've already heard some great context about why litter decomposition is important. Decomposition models are also important because they aim to integrate our empirical observations of the real world with process-based understanding, which allows us to do things like understand and quantify carbon and nutrient cycling by modelling their turnover from litter into the soil to predict nutrient availability and soil carbon sequestration, to predict future ecosystem responses to global change, as Mark's just highlighted, including how decomposition rates may change under future climate scenarios and impact terrestrial carbon flux, to identify key driving factors and separate their relative influences on rates of decomposition. And we can also use them to assess functional differences by evaluating how different ecosystems or plant functional types might contribute to decomposition dynamics. And perhaps most importantly, we can use them to inform some of our ecosystem management by providing predictions to guide land use and land management decisions, such as managing things like everything from agricultural residues to selecting species to enhance carbon storage. So, litter decomposition occurs in phases. It starts with an initial rapid early loss from leaching of soluble compounds and fragmentation, mostly by invertebrate fauna, over time periods of days to weeks. And then that's followed by sort of intermediate phase of more major litter fragmentation and microbial colonization and breakdown of more labile compounds over time frames of weeks to months. And then finally, there's a much slower late phase of microbial breakdown of more resistant compounds in the litter, such as lignin, over months to years. And our models of decomposition need to capture these observed dynamics. So the sort of established main controls on decomposition are typically climate, where warmer and to a point wetter conditions generally increase rates. Um, Environmental context, including things like soil type, oxygen concentrations or disturbance. Litter quality, which is measured in different ways, so things like carbon to nitrogen ratios or lignin content, which recognise, for example, that, say, soft, more nutrient-rich leaves will decompose faster than woody, lignin-rich material. And of course, soil microbes and invertebrate fauna are important. So our models of decomposition also need to capture the influence of these drivers. So I was just going to mention a recent global synthesis on some of the drivers of terrestrial litter decomposition, which found a big range in decomposition rates across the globe from polar to tropical regions, and found that litter quality and climate was explaining much of the variation in decomposition rates, and carbon to nitrogen ratio was identified as the best predictor in this analysis. So interestingly, soil biology came out as being of lesser importance, and I'll highlight its importance to modelling longer term soil organic matter a bit later on. And I just wanted to also mention that you can see from the map in the top left hand corner that Australia is relatively poorly represented in the total global data set. So, as part of a TERN funded project on greenhouse gas modelling, we've been reviewing some of the available data on litter quality, including carbon to nitrogen ratios and lignin content. And so far, our results are showing that there's quite a big range in carbon to nitrogen ratios in leaf litter and woody litter, which vary across different broad vegetation types. And significantly, many of these ratios are quite a lot higher than the global median defaults that we typically use in current models, which will make a difference to the sorts of predictions we make. And these new values are now being used to better inform some of our soil biogeochemical models in Australia. So I'll move on to a bit about some different types of ways of modelling decomposition. So models vary in their level of complexity. The most basic decomposition model is a single pool model, which incorporates the concept that litter decay follows what we call a single exponential decay function. So it assumes that the decomposition rate is proportional to remaining mass and is often informed by field litter bag studies. And we can quantify several metrics from these models. The mass remaining is the simplest. The decay constant, or K, summarizes the decay rate. And we can also calculate a half-life, which tells us how long litter takes to decompose by 50%. So this typically works well for many litter types, especially in the early stages of decomposition. And I've just got a figure to show here for Eucalyptus obliqua foliage mass loss in the first 2 years, showing quite a good agreement there with that simple model. Recently, we've compiled a database of observations on decomposition rates for Australia's native vegetation, and we've found that rates again vary quite a lot among not only the litter components of foliage, bark, and wood, but also some broad vegetation types. But notably, if you look at the distribution of observations in those maps on the left-hand side, there's a lack of representation for many vegetation categories and also components, particularly bark. So what we're doing at the moment is using the half-lives that we've calculated from these observations to calibrate some of our national carbon models. So we know that real systems are more complex than a single pool, and multi-pool models assume that plant litter is a mixture of materials with different decay rates. So in the two pool models illustrated here, there's a decomposable or fast pool consisting of more labile or soluble carbon compounds like sugars or amino acids that are easily broken down and have a high decomposition rate. And then a second, more resistant or slow pool that contains recalcitrant materials such as lignin or waxes, and it can also include non-lignified cellulose that resist rapid degradation and have significantly lower decomposition rates. And there are other models that can incorporate 3 or more pools as well. Um, Instead of looking at litter decomposition in isolation, most models treat litter as part of a continuous flow of carbon, ultimately resulting in either the release of CO2 to the atmosphere or as stabilization in soil organic matter. And this plant litter soil continuum links plant traits, litter inputs, and soil organic matter formation. And we can illustrate this flow from litter to soil, for example, using observations of isotopic labeling of litter to follow the movement of litter derived carbon from above ground litter into soil organic matter. And this is shown in this figure on the right hand side, showing the movement of litter derived carbon over time into the soil. And we can use these illustrations to inform our models. Um, so process-based litter decomposition models simulate the underlying biological, chemical, and physical mechanisms of organic matter breakdown rather than just fitting a curve to mass loss data. So several established models utilize multi-pool structures and use simple first-order decay rates. And these models are well suited for large long-term soil carbon predictions where microbial dynamics are relatively stable. Um, However, more recently, models have been advancing to improve how they represent the role of microorganisms in breaking down soil organic matter. So, by moving beyond sort of simple exponential decay models to capture some of the non-linear trajectories of decomposition, including things like the initial lag phases and slow late stage degradation. And these models include microbes explicitly and bring in enzyme kinetics and are better for understanding mechanisms and predicting responses to environmental change. And our team is currently working on 2 other microbially explicit models to the one shown here, one called MESC, another one called BAMS5 as part of our greenhouse gas modelling work. Some testing of these model advances towards more microbial realism have shown some similar performance between the microbial and conventional models for predicting litter decomposition, but substantial improvements for, and that's shown in the left-hand side, I should say, the observed versus predicted mass remaining, and the but substantial improvements for microbial over other conventional models for predicting soil organic has been found for predicting soil organic carbon, shown in the right-hand figure here, where we've got the conventional model highlighted in red and several versions of one of the microbially explicit models. And so, really, the emergence of a new paradigm which recognises that soil organic matter persistence is not determined by just how hard a molecule is to break down, but rather whether it's sufficiently processed by microbes and protected by the soil matrix. And that is driving advances in modelling the decomposition of litter and soil organic matter. So, just to summarise, litter decomposition is essential for carbon and nutrient cycling, and there's several controlling factors, and these need to be captured in our decomposition models. And as Heather and Mark have highlighted, these are likely to be potentially different in different ecosystem types. And we have new models developing towards microbial realism, but the processes are still quite simplified, and future work requires better integration of data and microbial detail. And importantly, we need more observations, more real data that are representative of a range of ecosystems to improve our model calibration and validation. So I'd like to finish with how we can apply these models. So there's been growing interest in measuring, monitoring, evaluation and reporting of greenhouse gases at different levels, actually. So model predictions can support estimates and reporting of these emissions at national and global scales. They can also inform land management and agriculture, for example, through supporting net zero emissions targets, and with vegetation, litter, and soil carbon playing a key role here. And importantly, they can also help predict ecosystem responses to climate change to guide proactive management. So just finish up with this slide, which summarises where we're heading with some of this modelling under a current TERN project on greenhouse gas modelling, where the project's testing 3 of these soil carbon models with increasing complexity from simple to microbially explicit. And these are being calibrated and tested using spatially distributed existing observations and new field measurements, with the ultimate aim to generate more robust predictions of soil carbon sequestration, among other things, in greenhouse gas emissions. Finally, I'd just like to gratefully acknowledge several contributors from CSIRO and our funders for funding the broader work in greenhouse gas modelling. Thank you.
Wonderful. Thank you so much, Jackie, and thank you, Heather and Mark, as well, for those really interesting talks. And we now have some time for some question and answers. So just a reminder to anyone watching, please either raise a hand or put a question in the question and answer box. One of the first things I was that we want to talk about is this one is for Heather. That's come through. How similar are the processes across different drylands in the world? Are some of the processes that you're seeing in the drylands in North America, for example, are we going to see similar things in outback Australia, or is it going to be very dependent on the different vegetation, different microbial communities?
Oh, that's a great question. Uh, I would I think there's definitely, I mean, dryland systems are so diverse, right? So we're lumping, almost half the terrestrial land area together here. Um, certainly there's going to be a lot of variability, and we have, I do some work in, hyper arid regions have been maybe like in really, really different processes there than like semi-arid systems in Australia or Arizona, right? So, um, and really different like litter chemistry and decomposer organisms. So I'd say like there's some unifying processes, right? So some similarity, and I think one of the really key things there is the spatial heterogeneity, right? The fact that these are that you don't have continuous canopies, but you have patches of plants and then these bare spaces in between, and that changes the dynamics. But we've looked at, yes, they've done some characterization of just like how stable are the surfaces, right? Are they, if the litter falls, is it likely to kind of stay in one place, or is it more likely to be moved around a lot? That seems to be one thing that does matter a lot.
So the landscape as well is playing a big role in that, and that's gonna be diverse in different dry lands as well. Okay.
Yeah.
And this one is, I think, for anyone to any of the speakers who want to jump in, a question that's come through is, what role do fungi play in recycling litter? Do some species speed up the rate of decomposition? And I suppose that touches on the discussions around microbial decomposition, how much of that is coming from fungi.
Do you want me to start off?
Go
Um,
for it.
sure. Yeah, look, I'm by no means a microbial expert, but yes, definitely, fungi will speed up decomposition. Particular groups of fungi will be able to target particular types of litter material and be more specialized. Some will be more specialized at, dealing with certain compounds than others. So, and I think, there's quite a lot of evidence to say that kind of the community, the mix of kind of microbial um, diversity also, plays a big role in that sort of rate of decomposition. But might hand over to Heather perhaps to cover off a bit more.
Yeah, I'll say basically the same thing. I'm totally, yeah, agreeing with what you said, Jackie. And I am also not a microbial expert, so yes, certainly, like fungi are super important, and that there's a lot of variation between systems on exactly who the most key decomposer organisms are. I don't know if Mark has things to add to that too.
No, you've done it nicely, both of you.
Wonderful. Thank you both. And this is a question for Jackie that's coming from Sean Walsh at the EECA. You mentioned a range of reasons why it's useful to monitor litter decomposition. Have you considered applications of your work to predicting litter fuel loads for fire modeling?
Yeah, okay, great question. Yes, look, I mean, obviously, one person's litter is another person's fuel. That depends on your perspective here, with a few, different definitions. But yeah, definitely, we so as part of our modeling in the broader sort of ecosystem level carbon accounting, we definitely consider, the consumption of fuel in our and following disturbances from fires and things like that. So definitely, we already have a little bit of a focus on that more around, sort of disturbance processes that affect some of these different pools. We also have colleagues at CSIRO who are through a big project called the National Bushfire Infrastructure Capability, I believe, that have done some recent modeling of fuel accumulation in different ecosystem types. So, there's a bit of crossover in those spaces. But yeah, great question. Definitely very well aligned.
Wonderful. Thank you. And so the next question that's come along is for Heather. With the upscaling work, how is the majority, if the majority of the litter is subcanopy, how do you propose to look at this from airborne or from satellites and other forms of remote sensing?
Excellent question. Yeah, this is definitely a challenge. As my remote sensing colleagues remind me frequently of, like, yeah, but we can't see the litter. But and they also assure me this is one of the one of the more challenging systems that they've worked in. But what we are finding is that we have pretty good relationships, or sort of our ground-based measurements suggest that we have pretty good relationships, uh, that we could fairly well predict where litter is distributed on the landscape if we make ground-based measurements of, say, canopy height, canopy size. So the plan is, and this is still very much a work in progress, but the plan is if we can use that remote sensing data to look at, say, the structure of plants. We won't be able to measure the litter underneath canopies, but can we get at least reasonable predictive capability to get an idea of what those pool sizes are?
We've just done a story in our newsletter about drone remote sensing. Is this an area where drones would be particularly useful, being able to capture a bit more of that lower altitude information?
Yeah, I mean, certainly they're if drones are getting low enough, well, still not low enough to go underneath the canopies, at least in our lower shrubs. Um, but certainly, whatever gets higher resolution imagery should help. We're working at the neon sites in the U.S. In particular because their airborne observing platform has very high resolution imagery flown from an airplane. Um, so yeah, but that it's going to be a challenge. We'll see how well we do at it, but um, hopefully improve our predictions of litter pools.
Wonderful, thank you. I'm just gonna get a question from Mark here to jump in. Mark, are the warming treatments altering the plant communities? And if so, how is this affecting the SOC and CO2 fluxes?
That also is a very good question, and yes, they do. So the warming chambers make the plants use more water because it's warmer, and they open their stomata more. They are active for more of the day, and what that does is it slightly dries the system, and other species, particular species, do better under those conditions of drier conditions. And so it means we have an increase in the grassiness of the grassland, if you will. And so a reduction in the cover of the other types of species. So the little herbaceous flat weeds, so things like daisies and dandelions and things like that. And also a reduction in sedges. And those are the ones that tend to have lower levels of decomposition and lower levels of respiration, and they actually use less water as well. So you do actually get this cycle where if you make it warmer, that makes it drier, that makes it grassier, which also makes it drier, which makes it grassier, which makes it drier, which makes it grassier.
It's a lot of cycles going on there. It gets very complex very quickly, it sounds. And then we've got a question about for Jackie, actually. Um, the person asks, I worked on leaf litter decay project in the Badongo forest in the Congo Basin from 2019 to 2020. This being a remote and demanding work. Jackie, are there works on how to minimize this field workload using modeling in remote sensing and GIS?
Okay. Yeah, that's another good question. So definitely we obviously, particularly in systems we don't know much about, obviously it's ideal to do some observations in the field. But yeah, I note that sounds like it's quite a challenging environment. Um, yeah, look, I think one of the things I say we can really apply a model to anything. It's just how well it works and predicts. So really, it's very difficult without some observations to test that. So in terms of remote sensing, that's not really my area of expertise, but we certainly, you know, more and more trying to link at least the pools of different, debris, biomass, and other things to remote sensing. So, yeah, I guess I probably defer on that one because it's not really, not really something we've looked at specifically around the litter pool.
Okay. Thank you so much. Look, unfortunately, that is all the time we have for questions. We do have a couple more questions. I will make sure that the speakers do receive those questions and are able to answer those for those who ask them. And I'm really grateful for the interaction and the interest that has been expressed here. It was a fascinating talk. Just going to share my screen quickly Just to let everybody know that there is there are going to be more webinars this year. We're very excited to. And so watch this space for the webinars that we will be presenting. The topics are to be determined, and we're always open to suggestions to see what particular areas you would like to see more discussions as robust as this one today about. So once again, I want to thank the speakers and thank everyone for your attention. I know it's a different time zones, especially Heather has stayed up late for us as she is in a very different time zone from us here in Brisbane. So thank you so much.